So welcome everybody. Thanks for watching us today. Today we're gonna have our lecture number 7 with Dr. Joren Madsen. Just to remind you that you can submit your questions by 3 ways. You can do it through the chat tab, or you can use Q&A tab, or you can hit the raise your hand button. You can find all these 3 options in the bottom of your screen. During this webinar, if you experience any technical problem, please contact me via chat. You can select my name, Thiago Borges, there, and just send me a message. And at the end of this lecture, I'm gonna send you feedback online. Please, if you can answer with your suggestions to help us to make this webinar better, that would be very appreciated. Now I'm gonna turn on to Leo, so he'll formally present Dr. Madsen today.
Thank you, Thiago. It is my pleasure to introduce Dr. Joren Madsen as the 7th speaker of our series. Dr. Madsen is the director of the Mass General Transplant Center, the co-director of the Center of Transplantation Science, and the Paul Russell Warner Lambert Professor of Surgery at Harvard Medical School and Professor of Surgery at MGH. Today's webinar is entitled The Innate Immunity in Transplantation, and now I'll turn it over to Dr. Madsen to begin today's webinar. Thank you.
Thanks, Leo. Thanks, Theo, and also thanks to Sandro for organizing this summer lecture series, which have really been fantastic and has raised the bar on these informational sessions that I think everybody really appreciates. Today, I'm going to talk about innate immunity in transplantation, which is something that is relatively new to transplanters. As I'll mention later in the talk, historically, our focus has been on T and B cells, and it's taken a while for us to realize as a community that the innate immune system also is an important factor in our transplants. And my aim today is try to convince you of that. So the second thing I wanted to mention is that the way I've organized this talk is to really couch the information and educational aspects in data that has been generated in the CTS by your fellow investigators. And that way we can not only learn about innate immunity, but learn about some of the studies that have gone on before some of the newer people have arrived in the laboratory. So with no further ado, now I guess my slide isn't moving forward here. Let's see. There we go. So the innate immune system was evolutionarily the first defense mechanism that organisms had to combat microorganisms, microbes. And it really began, people think, around 700 million years ago that the first systems were developed to do 2 things. One is to prevent invasion of microbes and etc. And the other is to clean up or mop up damaged cells and dead cells. And then about 450 million years ago, the more adaptive type of immune system we all recognize today developed first in jawed fishes and then recently in human beings. If anybody is interested in the immunology of fish, Sandro has some interesting tidbits about the immune system of fish and how it relates to kidneys. The cells of the innate immune system are here. I will talk about the ones that are circled. It's important to realize that there's more to the innate immune system than these cells. The complement system, skin itself, are all part of the what we call the innate or natural immune system. So everybody knows that one of the defining features of the innate immune system is the timing of activity. Whereas T and B cells take days to weeks to become activated and to develop a response, the innate immune cells take hours. And it's a quick on and a quick off situation. And this slide from Christian Flock, this really nicely shows this comparison. I want to spend some time on the receptors and ligands of the innate immune system. So this was developed in a very elegant way. The receptors, which are called toll-like receptors, toll because they are homologous to a protein in Drosophila, which is a toll protein that also seems to protect the flies from invasion by infectious agents. Toll receptors are part of a set of pattern recognition receptors. So there's at least 9 toll-like receptors, which are a subset of PRRs. What do they recognize? They recognize 2 different types of patterns, molecular patterns, and those are patterns that relate to pathogens, infectious disease agents, and those that relate to damaged cells. It's very interesting that these receptors are specific for either bits of bacteria, bits of fungus, bits of viruses, or components of cells that leak out during necrosis, apoptosis, and are inflammatory. So in terms of, let's start with the pathogen-associated molecular patterns. So for instance, when a gram-negative bacteria invades, part of its cell wall is made up of LPS. There's a specific receptor on innate immune cells called TLR4 that's specific for that LPS protein. Likewise, HMGB, which is a histone protein, is released on cell death, and there's another TLR protein, in this case TLR9 and TLR4, which is specific for that protein and activates the innate immune system. What the key point of this system is that none of these proteins, glycoproteins, DNA strands, are present in the host. So these are all specific to the agents that are potentially causing damage. And that's really the basis for the recognition pathways in the innate immune system. And this is, in the cartoon, a pathogen produces PAMP, which is seen by PRR, which is, as I mentioned before, a subset, which is toll-like receptors. And this is an APC, which I'll explain later. And then this APC then talks to the T cell and starts the process of adaptive immunity. And in the early days, evolutionarily, it would just be the APC that was responding without this component, resulting in inflammation and elimination of the damaging or potentially damaging material. So comparing the innate and adaptive immune systems receptors, there are about 100 types of innate immune receptors, either the PRRs, that recognize about 1,000 different PAMPs or DAMPs. So these receptors are encoded by the germline. So these cells, those cells of the same lineage, have identical receptors. This is an important point. So all the macrophages, all the dendritic cells, all the NK cells have the same type of receptor for these PAMPs and DAMPs. This is in contradistinction to the adaptive immune system, whereas there are really only 2 major immune receptors, the TCR and immunoglobulin, or the BCR, but they can recognize millions and millions of different antigens. And this is, is because of the fact that these genes or gene segments undergo somatic rearrangement during lymphocyte development, which results in a huge amount of diversity. So this is a bit of comparison of the 2 immune systems. Now, all that being said, there are some unique examples that will, if you will, contradict this paradigm that has been set up. And one of them is shown here, where a transplant situation in sponges or corals elicits a rejection response. Now, theoretically, these sponges or corals shouldn't be recognizing a separate member, if you will, of its phylum. And it shouldn't be able to mount a rejection response in this situation. So there's something else going on. And this was actually brought to the fore recently by Fadilakis's group, where they've shown that monocytes have the capacity to recognize non-self and actually induce an allograft rejection. So this is not against a microbe. This is not against damaged cells. This is an allograft rejection. Again, doesn't really fit in the paradigm I just mentioned to you. And throughout this talk, I'm going to be throwing little monkey wrenches into the historical conventional wisdom. This is one. How this happens, we don't know yet, but it could be important. And I'll get back to this later at the end of the talk. So this is the classical example, if you will, of how the innate immune system works relative to a transplant. We have the surgical procedure that produces tissue damage. Usually these organs, for the most part, come from brain-dead donors. That lends itself a state of inflammation that releases damaging elements and damps, if you will. We have ischemia reperfusion injury, which also is a factor in damaging cells. And it could be translocation in some organs of bacteria. So we have a setup for the production of DAMPs and PAMPs, which then begin the process of activating, in this case, macrophages, but it could be other cell types too. The one bit of information just to prove that DAMPs are actually important. This is a mouse study in which the investigators use an anti-HMGB1 antibody to show that by preventing the activation of innate immune cells by this DAMP, they were able to prevent chronic rejection or cardiac allograft vasculopathy in a mouse model. And you can see here the control antibody versus the antibody against the HMGB1, quite a difference. And as the talk goes along, I might forget to mention which species I'm talking about. If I do, just look up in the right-hand corner and it will let you know. Our laboratory, in fact, Jim Allen's laboratory, also has shown the importance of the innate immune system in organ transplantation. This is a orthotopic pig lung transplant model, which Jim has used over the years, and has shown that with a short course of cyclosporin alone, 12 days actually, most organs from a non-brain dead donor go on long term. They become tolerant. However, if you induce brain death in the donor prior to procurement, and then you keep the organ, the donor or the donor on a ventilator for 4 hours, and then procure the organ, you see that there is acute rejection in every single case. So this makes it clear that there is a component of innate immunity playing a role in the ability of this protocol to induce tolerance. This is substantiated by looking at the tissue cytokine expression, and you can see that after 4 hours of brain death, you see an increase in the production or expression, if you will, of IL-1 and IL-6 in this case. These are cytokines associated with innate immunity. Interestingly, and I'll get back to this later, there are organ-specific differences in how the different tissues respond to brain death. You can see in the kidney situation, we see very little increase in these deleterious cytokines versus the heart, where you see even greater increases in IL-6 and in this case, interferon gamma. Point is that brain death does significantly impact tolerance, at least for some organs, and this is through innate mechanisms. So the first cell we'll talk about, dendritic cells. These are also referred to antigen-presenting cells, or APCs. They're also historically referred to as passenger leukocytes, and you may read that in your earlier literature. When they're talking about passenger leukocytes, they're talking about dendritic cells. I consider the dendritic cells to be the king of the innate immune system. They're 100 times more effective than macrophages as APCs. They're found in lymphoid and non-lymphoid tissues. They're relatively rare, and like so many cells in both the innate and the adaptive immune system, the phenotype and function of these cells is modulated based on the maturity of the cells, the tissue location, and the milieu in which these cells are found. Importantly, though, for this talk, the dendritic cell really is the ultimate bridge between the innate and the adaptive immune systems. So I want to demonstrate that here. This is, I think, more of a macrophage than a dendritic cell, but it's a good cartoon because it really is clear how this process develops. With the APC recognizing a PAMP, in this case, becoming activated, maturing, and then being able to present antigen to the T cell. Okay, this is a classical paradigm for how the innate and the adaptive systems relate. And this is a dendritic cell, and this is getting along more toward the talk that Marissa gave last week or 2 weeks ago, and reminding you that there are 3 signals. I won't go into the details, but the signals include antigen presentation, co-stimulation, and cytokine production. And this is what is necessary for complete activation of the T cell by an APC. So again, this is what you will read in the immunology textbooks. This is what you'll be taught in Immunology 101. And it is a foundation for what we call the passenger leukocyte theory of allorecognition in transplantation. And again, this is conventional wisdom. This is what we have accepted as gospel for the last, well, 30 to 40 years. And in essence, the theory is that donor dendritic cells activated by DAMPs or PAMPs migrate to draining lymph nodes or the spleen, depending on the organ transplanted, of the recipient, and they present the donor MHC, the dendritic cells, complete whole MHC molecules to recipient T cells, and this is called direct allorecognition. This is what Marissa talked about. And those T cells then traffic back to the organ as cytotoxic effectors causing graft rejection, and is all well spelled out here. Again, classical training. I raise this because it could be all wrong. And the reason it could be all wrong is because of recent work showing that there is another pathway using a process of exosome generation, vesicle production, that can carry donor MHC molecules to the recipient cell, and these exosomes become integrated into the membrane of the recipient cell. And you have a situation where the recipient, in this case, a recipient dendritic cell, is expressing both the recipient or host MHC and the donor MHC that it has acquired through cross-dressing, the process of exosomes integrating into the cell membrane of a different cell, and that those proteins being expressed on the cell surface, cross-dressing. And this was the foundation for a really very seminal experiment that Gilles Bénichou published in 2016. And the reason I bring it up, because during Marissa's talk, it really wasn't, I didn't think, given enough showtime. I mean, it didn't, I don't think there was enough emphasis placed on the importance of this concept. The concept is that perhaps the recipient dendritic cell is more important in transplant allorecognition than the donor dendritic cell. And although that's the beginning of the experiments performed by Gilles, I think it encapsulates the point. And in this case, I'll bring your attention to this section of the slide, heart transplants, days after grafting. And this is looking at either donor leukocytes, okay, dendritic cells, or cross-dressed cells in draining lymph nodes, non-draining lymph nodes, and spleens. And grossly, there was quite a difference. In terms of the lymph nodes, there's an order of magnitude difference between the two cell types. And in the spleen, there were multiple orders of magnitude difference between the two types of cells, the donors versus the recipients that were cross-dressed. He goes on to prove, I think, that recipient antigen-presenting cells cross-dressed with donor MHC rather than passenger leukocytes, dendritic cells of the donor, trigger T-cell responses after allotransplantation. As I see it, this means that recipient DCs, as opposed to donor DCs, are increasingly recognized to be the key players in allorecognition. And this is something called semi-direct presentation. So I just wanted to bring that to your attention. So how else are dendritic cells important to our work? Well, dendritic cells play a key role in self-tolerance. Here's a thymus of a young child. It's nice and big. And again, I won't go through the details, but dendritic cells traffic from the blood to the thymus, where they partake in two important functions. One is clonal deletion. And this is an example of a cell, a thymocyte, a T-cell precursor who has a high affinity receptor for a self-MHC molecule and could potentially cause autoimmunity if it were released into the circulation. So it undergoes clonal deletion, and the dendritic cell is an important participant in that process. It also is an important participant in the production of regulatory T cells. So there's really two functions that are encompassed by dendritic cells in the thymus. Why is that important? Well, I'll show you. This is just a cartoon to reemphasize the importance of the dendritic cell. T cell precursors migrate to the thymus. They undergo positive and negative selection, again, as I mentioned, through dendritic cells. And they come out the other side as educated to self and as self-specific Tregs, which are, I guess, teleologically present to help mop up those T cells that weren't educated correctly in the thymus graduate school. And then these cells then migrate to the periphery. So what about mixed chimerism? So there's a lot of work in mixed chimerism going on in the lab, and one of the hypotheses that underlies the success of this protocol, again, involves dendritic cells. We provide a donor bone marrow transplant. That marrow includes dendritic cells. These cells traffic to the thymus, where now there are two teachers, if you will, in the thymus. There's the dendritic cells from the host and dendritic cells from the donor, and they work together theoretically to educate maturing T cells to see host and donor as non-foreign, and to produce Tregs that are specific for self and donor, again, to tamp down any autoreactive, or in this case, donor-reactive T cells that escape the thymus. So another way dendritic cells play an important role in our work. Another important point about dendritic cells is that the function of them depends on the maturity of the cell. Immature dendritic cells are thought to be more tolerogenic, if you will. They haven't been shown to promote regulation by promoting Tregs, in contrast to mature dendritic cells, which are mainly the antigen-presenting cells that promote the Th1, Th2, and Th17 responses. I'll get back to the importance of this distinction later. We'll move on to plasmacytoid dendritic cells. This is a subset of DCs. This is one of Sandro's favorite cells. They are so named because they resemble plasma cells, very rare in the bone marrow and in the peripheral blood, very rare. Their main function in human beings is to produce type 1 interferons to fight off viruses. That's their real major function. They're less efficient than DCs in presenting antigen T cells, but the take-home message here is that they are very effective at generating Tregs, and that's partly because of the fact they express PD-L1, and they produce IL-10 and IDO, which again are important for Treg generation. So we have found an interesting phenomenon that Sandra really should get credit for, and this will take about a few minutes to describe. And it goes way, way back to, I think, the 60s, where Dr. Paul Russell and Dr. Bob Colvin made a very interesting discovery. When transplanting kidney allografts in mice, they found that in some strain combinations, these kidney allografts were spontaneously accepted. So the DBA to BLAC6 strain combination results in kidney allograft survival in untreated recipients, spontaneous acceptance. If you reverse the strain combination, you don't see tolerance. In fact, they are all rejected. Furthermore, if you take the heart and transplant that instead of the kidney in the same tolerogenic strain combination, you see that the heart's rejected quickly. So there's quite a organ-specific component to this process or phenomenon of spontaneous tolerance. There's two things I just want to show you. One is that Sandro has shown that the plasmacytoid dendritic cells are, the load of those cells in organs differs. In kidneys, we see a number, the presence of these cells quite well using the PDCA1 marker, whereas you don't see them in hearts. I don't have time to show you all the data in this paper that he just published, but what he also found was that there was a strain-specific difference in the ability of PDCs to induce Treg generation. And this difference was correlated to the differences in the induction of, if you will, spontaneous tolerance between these B6 to BDBA and DBA to B6. So in essence, the DBAs were able to, the PDCs from the DBAs were able to induce Tregs more potently than the B6. So a way that PDCs may be playing a role in this particular phenomenon. Where do the PDCs seem to hang out? Well, they hang out in a unique lymphoid structure that Bob Colvin has termed Treg-rich organized lymphoid structures, or TOLS. And so this is a kidney from one of these animals that underwent spontaneous acceptance of a DBA kidney. And these are the TOLS very clearly outlined here. And these TOLS, if you do immunohistochemistry, are chock full of Tregs, but they also contain PDCs to a fair degree. So again, could be an important mechanism of this phenomenon. Likewise, they may be an important participant in another phenomenon, and this is another tolerance model, but this is active tolerance. This is not spontaneous. This is a mixed chimerism-induced tolerance. And as most of you know, there's a organ-specific difference in the ability of mixed chimerism to induce tolerance of a kidney versus a heart allograft. This is true in pigs, this is true in mice, and it's true in monkeys. We've also shown that if you combine the heart and kidney, the kidney can confer unresponsiveness upon the heart. And if you look at the kidneys in both situations, you will find TOLs, and you'll find PDCs in those TOLs. So we are in the process of doing an experiment to deplete PDCs in these heart kidney recipients to see if we can aggregate tolerance. We haven't been able to do it because there's been no antibody that has been able to target PDCs and deplete them in monkeys, but we're in the process of characterizing one as we speak, and Parth is actively pursuing that line of investigation. I should just mention that in the tolerant human beings that Ben and Tatsuo and David Sachs have worked to find actually produce, if you will. We also find TOLS. I don't think we've actually stained for PDCs in those recipients yet, but TOLS seem to be a common thread in kidney tolerance induction. I'll just end PDC story by showing that if you adoptively transfer PDCs, you will normally find enhanced graft survival, and if you deplete PDCs, you abrogate tolerance. So PDCs, I think, are important, and you'll probably be hearing more about them as we learn more about them. On to natural killer cells. This is one of my favorites. We have been interested in natural killer cells for a number of years now. They make up 10% of the PBMC. Their primary function is destroying virus-infected cells and tumor cells. They do that by producing interferon gamma, TNF, alpha, perforin, granzyme B. Importantly, they participate in antibody-dependent cell-mediated cytotoxicity, or ADCC, via their CD16 receptor, which binds the FC receptors and antibodies. The NK cell has a very interesting inhibitory receptor. And so this receptor is activated by the absence of a molecule, specifically a self-MHC class 1. So if the NK cell is presented with self-class 1, the inhibitory receptor works to prevent activation of the NK cell. But if that class 1 molecule is not present, this will induce activation of the NK cell. This is called the missing self hypothesis. As I think about it, T cells destroy other cells for carrying foreign class 1 MHC, while NK cells destroy other cells for not carrying self-class 1 MHC. They play a dominant role in bone marrow transplantation and xenotransplantation, less in allograft rejection. I'll get to that in a minute. And they're part of a new spectrum of lymphoid cells that are termed innate lymphoid cells, or ILCs. So on the one hand, NK cells really, I don't believe, can reject allografts on their own. But this data shows that they can participate in allograft rejection. And real quickly, this is a heart transplant model, and it's using recipients that are CD28 knockouts. If you look just at these two slides here, this is a recipient of a heart into a CD28 knockout with a control antibody. You see the median survival here. But if you actually give an antibody that depletes NK cells, which is anti-NK1.1, you see prolongation of graft survival, suggesting that the NK cells can provide help to the T cells that are rejecting the allograft in a CD8 deficient host. And this has been substantiated by other studies, by Marissa Allegre. So that's on one side of the coin. On the other side of the coin is data from Ron Gill's lab on the left and actually Jonathan Bromberg's lab on the right, showing that NK cells are necessary for tolerance induction, at least tolerance induced by co-stimulatory blockade. So this is an islet cell model, and just to be brief, this is animals that were treated with the CD154 monoclonal antibody. They see prolonged graft survival. And then if you give the CD154 and the anti-NK1.1, you see a diminution in allograft survival. Jonathan did this very similar experiment using anti-CD40 ligand and a DST model. And again, The control antibody with the type triangles go on long term, but if you add NK1.1, you see abrogation of tolerance. So two sides of the coin that are called NK cells. Well, how does that affect us? Well, again, getting back to Dr. Russell's very seminal early experiments, he and Bob Colvin published a paper in 2001, which is very interesting, and I'm going to take some time to go over it. This is a mouse heart and skin transplant model. Two types of tolerance were induced: neonatal tolerance and mixed chimerism. Now, there's no one better to study neonatal tolerance than Dr. Russell, who worked with Peter Medawar, who was there at the very beginning. And in the paradigm here is that they induced tolerance, and then they did skin grafts that lasted long term. The skin grafts were just to prove that tolerance had been achieved. And this was also proven by the fact that there were still donor cells circulating in the recipient. The real experiment was then to transplant a heart challenge graft. So these hearts were from the same donors as the skin. And although these hearts went on and survived long term, when they actually took the hearts out and studied them, they all had severe, if you will, cardiac allograft vasculopathy. So it was a strange, counterintuitive situation where you have a classical model or system of tolerance, but you see a severe response in the vasculature. And we were trying to understand how that could happen. And there were a number of indicators that suggest NK cells might be a reason. So we developed a very non-contrived model to study NK cells, and that's a parental to F1 model. So the parental F1 model uses B6, which are black, valve Cs, which are white, and when you mate them, they develop a beige coloration. Now, everybody out there realizes that if you transplanted a skin or a heart from either of these mice into the F1, they will be accepted long term, because these mice are educated to both H2B black and H2D white. However, the NK cells will respond to either parent, and I'm just going to take a moment to describe why. This is something called hybrid resistance, and you won't understand the experiment unless you really understand how it works. And this is an important, issue in parental to child bone marrow transplantation. And so the way it works is that the F1 recipients gets two buckets of NK cells. They get the NK cells from the white mouse, and they get the NK cells from the black mouse. And I have demonstrated or designated that as white here and black here. So the NK cells from the white mouse see parent A, which is the white mouse, and are not activated because of the inhibitory receptors. However, if they were transplanted with a heart from a black mouse, there will be no self-MHC class 1, and it will activate the NK cell. The same is true for the segment of NK cells that are from the black. The same thing happens. If you transplanted a organ from the black mouse, nothing will, no effect, inhibition. If you transplanted it from the white mouse, you'll see NK cell activation. And the key here is that no matter which parent you use, there's going to be one cell type, one NK cell type that's going to be activated. So that's the basis for the experiment. And it's a sweet model because of the fact we don't have to use antibodies or knockouts to really isolate the effects of the NK cells. And this is a long-winded way to show you the results. And I'll start with the controls. So the F1 to F1, there was no demonstration of CAV on histology. If we do the black 6 into the black 6, syngeneic, no CAV. However, if you do the black 6 into the F1, pretty much every heart develops severe CAV, suggesting strongly that NK cells are playing a role. And in fact, we looked at NK cell cytotoxicity and proved that it was present at least up until day 35. As the lesions matured, they matured even after the cessation of NK cell activity, but clearly this is a process that could be responsible for that development of this damaging immune process in a recipient that's otherwise tolerant. We did more studies using NK cells, and I think one of the other important findings was a novel pathway of antibody-mediated CAV. And again, this is work that was published in 2012 with the Bob Colvin, Paul Russell, Sandro group. And I won't go through all the details, but it's a very interesting finding, especially by comparison to more newer data, looking at transcripts from humans that are undergoing antibody-mediated rejection, and noting that there are significant populations of NK cells and NK cell transcripts in these biopsies, which correlate with the findings that we produced in the mouse. And again, I won't have time to go through all the data. But to summarize the NK cell story, I'll just take you through this real quick. The NK cells have the ability to induce antibody ADCC through their FC and CD16 interaction. They produce interferon gamma, which can enhance T-cell mediated rejection. And on the other hand, they can produce an IL-10, which may affect and promote Treg and regulation. So it's a complex story, again, dependent on, as I mentioned, the maturation, the cytokine milieu, and the tissue these cells are in. Interferon gamma. So if DCs are the king of innate immunity, interferon gamma is the queen. It's pretty much involved in at the effector end of all innate immune cells. And we actually asked the question, could NK cells in our model be working through an interferon gamma mechanism? And the answer is yes. We did this by using interferon gamma knockouts and showed that if the recipient is an interferon gamma knockout, the F1 recipient, we see no CAV. However, if we used knockouts of the interferon gamma cytokine or the receptor in the donor, we didn't see any change in CAV. So this is a recipient-specific process. And this just shows the difference in the neo-intimal index between the knockouts and the wild type. And again, won't go into the details. This is an impressive publication by a friend of mine who was in Oxford with me, George Chalides, who is a cardiac surgeon at Yale. And he showed that interferon gamma alone, in the absence of leukocytes, can induce intimal proliferation in both pigs and in mice, which was, I thought, a pretty striking finding. We read this and we started thinking about interferon, and were interested in asking questions about it. So we developed a model in which we were able to continuously infuse interferon gamma into pig hearts before they were heterotopically transplanted. And Paul Russell would call this a real surgical experiment. It was a very interesting set of studies. So in brief, the catheter was placed to the aortic wall into the coronary artery halfway down. It was secured. It was then attached to an osmotic pump, and it allowed us to continuously infuse porcine interferon gamma throughout the course of the experiment. the pump was attached to the back of the pig, and it worked. So I'll take you through the results. This is the treatment group, the animal number, and this is histology at various weeks, the graft survival time, the mean survival time. So if we transplanted pig hearts across a MHC class 1 barrier, they all reject in 7 days if they're not treated. If we're given cyclosporin, they go on to average of 46 days. As I mentioned before, like the monkey, pig hearts do not become tolerant alone. If you administer saline through this mechanism, you don't really see a significant difference. But if you infuse interferon gamma, you see a stark diminution in allograft survival time. Not surprising. Then we asked the question, what about syngeneic and basically autologous hearts? So this is a situation, one of the unique aspects of the pig colony is we can inbreed to very high levels of consanguinity. And these are G7 versus G8 hearts. The G7 hearts we transplanted without therapy, without treatment, and they all went on long term. In this case, we took an even more, the next generation, the G8 heart, and infused interferon gamma and look at how quickly they rejected. Interestingly, when we actually did this in the native heart, and this required bypass, so we put the animal on bypass, clamped the aorta, opened it up, stuck in the catheter, and infused interferon gamma, there was no effect on the native heart. So just this small amount of difference was enough to cause rejection in the presence of interferon gamma. The real kicker experiment was this. We compared heart kidney recipients, which, as I mentioned in the monkeys, induce a result in allograft tolerance. In the pig experimentation, the same thing happens with a short course of cyclosporin. And if we give nothing, these animals go on long term. And if we infuse interferon gamma, the exact same protocol, all these animals went on long term. They didn't reject, which I thought was quite dramatic, suggesting that tolerance induction can can occur in the presence of elevated interferon gamma levels, and that tolerance induction can prevent the detrimental local and systemic effects of interferon gamma, which bodes well for tolerance and innate immunity. Interestingly, the long-term survivors did develop CAV, as George Toledes would have predicted, compared to the non-interferon gamma treated animals. So I just end the interferon gamma part of the talk by throwing another wrench into your thinking, and this is work by Catherine Wood at Oxford, showing that in essence, in a model where Tregs are adoptively transferred into a skin-grafted recipient and produce long-term graft survival, if you added a anti-interferon antibody to diminish the effects of interferon gamma, you see, again, abrogation of tolerance, demonstrating that in certain circumstances, interferon gamma is necessary for regulation. So confusing, but real. So the final part of the talk is about macrophages, and they are, I would say, in the spectrum of innate immunity, the most overlooked when it comes to transplantation, even though they make up a large percent of the rejection histology you see in all the different types of organs undergoing acute rejection. They were studied by William Kitchens, who now is a transplant surgeon at Emory, in our parental F1 model. I think part of the reason why macrophages aren't studied more in transplantation is just it's very hard to deplete macrophages. One way to do it is to use carrageenan, which is a very inflammatory reagent from seaweed. It causes thrombosis, and the only way we were able to do this experiment was to give aspirin along with the carrageenan. But we were able to show that by giving this macrophage depleter, we saw a diminution in the number of animals that developed CAB. So that also seemed to be playing a role in this process. The important take-home message about macrophages are that there are two types, and there are probably more than two types, but for now, the conventional wisdom is that there's an M1 and an M2, and basically the M1 is an alloregressive type of macrophage. The M2 is more of a tolerogenic type of macrophage. I'm not going to go into the details of this. But what I wanted to end the talk on, again, in the macrophage section, is to talk about memory. Again, conventional wisdom, innate immune system, no memory, adaptive memory. However, there are reasons to believe that this may not be completely correct. And this is a paper from 1977 showing, again, a coral transplant model. And they were able to demonstrate that, actually it's not a transplant model, it's a reaction to a particular microbe. The take-home message is that they were actually able to demonstrate a second set response in these corals that one would not have anticipated in a situation where memory did not exist. Second set response is, as you know, the more exaggerated response that is seen in T and B cells after a first initial contact with an antigen. So this is an interesting finding, and it leads to the end of the end of the talk, and that is just a glimpse of a new phenomenon called trained immunity. And I didn't include the data, but I just wanted to summarize it for you since it's at the end of the talk. And the concept is that macrophages activated by DAMPs, which can include the HMGB1 and vimentin. I was going to show more work actually from Agnes and her and Robin's work with vimentin, but that vimentin is something that can be considered an activating agent for innate immunity. This the production of these DAMPs during transplantation or following transplantation leads to a temporary change or reprogramming in the epigenetic and the metabolic processes of myeloid cells. And this is a process that has been referred now to as training. I won't go into the details of what that is, but I can happy to talk about that afterwards. These quote-unquote trained macrophages have a heightened inflammatory response upon re-exposure to donor graft antigen, and consequently induce a more potent adaptive alloresponse. And work by Jordi Orchando at Mount Sinai has shown that therapeutics that block this epigenetic rewiring and these metabolic modifications that underline trained immunity promotes M2 type of macrophage and can actually prolong cardiac allograft survival in mice. So this is something that we are interested in, and we're working with Jordi and others at Mount Sinai to look at in our mixed chimerism models. And I throw this out there. If you want to learn more about this, this is an article we published. It made the cover of the January issue of Transplantation. So you'll learn more about trained immunity there. So I'll end the talk with a summary conclusions. These are just kind of global takeaways. First is that the boundary between innate and adaptive immunity is becoming blurred. I mentioned that in regards to memory, allorecognition, and the ILCs we talked about. Like all immunocytes, the phenotype and function of cells varies dramatically based on the cell maturation, the tissue localization, and the cytokine milieu, going from alloaggressive to tolerogenic. Emerging data shows that the damage from early innate immunity has significant deleterious effects, both in the short term and the long term. following transplantation. Importantly, modern immunosuppression really has been focused on controlling T-cell mediated responses. However, since innate immunity seems to play a critical role in allograft survival, really to achieve acceptable long-term graft survival and patient survival, and even tolerance, we're going to have to start addressing innate immunity. The induction of tolerance may prevent or mitigate the damaging effects of both adaptive and innate immunity. That's based on our interferon gamma studies, which is, again, bodes well for studying tolerance in a setting of innate immunity. And finally, clinical trials using some of these regulatory innate immune cells, the dendritic cells we talked about, the macrophages, the M2 macrophages, are proceeding in the one study, something that Jim Markman has participated in. So that's it on the innate immune system. I hope you've enjoyed that and learned something about that immune system and a little bit about the work that's gone on in the CTS. Thanks.